So today we are going to discuss the muscles, especially the skeletal muscles. Muscles cannot push, they just pull. And with the help of pulling, they can help you in doing the push-ups, pull-ups, chin-ups, walking, sitting, running, jogging, all the activities of daily life. That's basically due to the help with the help of muscles. Out of all the weight of human body, about 50% is due to the muscles. There are three types of muscles: skeletal muscles, cardiac muscles, and smooth muscles. The skeletal muscles, they contribute almost 40% of the weight of a human body, while the rest of the weight is basically due to the rest of the 10% weight is basically due to the smooth and cardiac muscles. We in the last few lectures we have been discussing that we have been discussing the membrane potential and the action potential, and we discussed that there are only two types of cells which are excitable or which can pass current, and one of those were the nerve cell and the other is the muscle cell. So we discussed the action potential, but we did not discuss the how the action potential help in the movement of muscles, and then how the movement of muscles help us in executing the daily activities of life, like drinking, eating, walking, running, etc. Then the action potentials, basically they are signals that come from the nervous system, and with the help of neuron they enter the muscles, and then they help contract the muscles, and with the help of contraction of different groups of muscles in the upper arm and lower upper limb and lower limb, we can do different activities of our daily life. So how the nervous system interacts, how they both coordinate to execute a proper function of a daily life, we are going to discuss that in detail. And to before going into detail, we must study the structure of the muscle at the molecular level, as well as its basic anatomy, the anatomy of the muscles. If you look at the muscle, we cut the muscle in transversely into two parts. Suppose, for example, this is a whole muscle, and this muscle has been cut into two parts. We are not having the other part, and we are just looking at the one part. So this muscle has nicely, beautifully cut into two parts, and this one part is attached with this bone. This is the bone. This is the tendon, and the muscle is basically attached with the bone with the help of a tendon. So when we look into this muscle, this is basically made of three bundles. There are three pockets. These pockets basically contain muscle fibers. These pockets contain muscle fibers, but each pocket is known as a fasciculus. If the muscle as a whole is a grandfather, the fasciculus is father, and the muscle fiber is basically the son. So grandfather, father, and son. So the muscle fibers, they make pockets. These pockets are known as the fasciculus or fasciculi, and they basically contribute the whole of muscle. On the top of the muscle, there is a layer of connective tissue that is known as epimysium. So there is a connective tissue that is known as the epimysium. Inside the muscle, there is connective tissue that is surrounding the fasciculi. Or this is, if we have enlarged this one fasciculus here, this is basically one fasciculus which is made of a lot of muscle fibers. So the whole muscle is surrounded by epimysium. The fasciculus is surrounded by a connective tissue known as perimysium, and then the muscle fibers, they are surrounded by endomysium. So perimysium on the outside, epimysium on the fasciculi, and endomysium on the muscle fiber. The endomysium, they are continuous. They are basically thick connective tissue. They are continuous. they get attached with the they can just get attached with the perimysium, that is connective tissue above the fasciculi, and then that connect with the connective tissue on top of the muscle, that is the epimysium. So the epimysium, the perimysium, and the endomysium, they all unite together to form a tendon. This part of the muscle is known as tendon, and this is basically connective tissue, and that has been formed by the union of the epi, and endomysium. If we look at the detail of muscle fibers under a microscope, the muscle is formed by fasciculi. Fasciculi are formed by muscle fibers, and the muscle fibers, they are basically made of myofibrils. So they are made of multiple myofibrils. So each muscle fiber, it is basically made of multiple myofibrils, and these myofibrils, when seen under an electron microscope, not an ordinary microscope, they are having dark and light bands. All the muscle fibers, which are basically made of myofibrils, they are basically made of dark and light bands, and this structure is visible under the electron microscope. This is the basic functional unit of a cell, of a muscle. This is the main cell of the muscle, which in abundance form the fibers in the fasciculi and the whole muscles. So anything happening over here will get transferred to the fiber and the fasciculus and the muscle, and will manifest in the form of movement. So any movement of the finger, for example here, it is basically at the level of the myofibrils, at the level of the dark and light bands. These dark and light bands are due to different types of proteins. The dark band is basically known as the A band, and the light band is basically known as the I band. A band because it's anisotropic, and I band because it's isotropic. Isotropic is anything through which when light comes, it gets reflected at its own angle. The angle at which it's gets reflected with the same angle. But anisotropic means when light comes into it, not only get reflected at this angle, it can scattered into different directions. So this is just a property of light, and that property has been taken and used in the naming of the muscle fibers, the light and the dark band. So the dark band is basically the A band, and the light band is basically the I band. These dark and light bands, they are the functional units. They are the functional units of the muscle. If we look into this dark and light band into detail, it will look like this. And between a light band, there is a Z line, and the distance between two Z lines is known as sarcomere. The distance between two Z lines is known as sarcomere. So the region, the space from this to this line is known as sarcomere. And the protoplasm of this sarcomere is known as sarcoplasm. So myofibrils, they are made of dark and light bands. the light, there is a band, there is a line, or the known as the Z line between the I band. The I band is basically divided into two parts with the help of this Z line. This is known as Z line because either its appearance is like a Z shape, or some books says that the word, the alphabet Z has been taken from some German word which I cannot even pronounce. So that's not important. The concept which is important to understand is that the I band has been divided into two parts. That is Z the Z line. With the help of the Z line, this this Z line is basically made of special type of proteins. If we enlarge this A band, we will see that this A band is having some thick molecules. This is having some thick molecules that are known as the myosins. And this, if this I band is enlarged, this is basically made of some molecules known as actins. So these are basically the molecules, the myosin and the actin, which basically are forming the I band and the A band. The I band is contributing a small part towards the sarcomere from both sides. There is one I band on this side of the A band, there is one I band on this side of the A band. So one small portion of the I band from this side and one small portion of the I band from this side, they come along with the A band and form the sarcomere. And this is the functional unit of this. This is basically, this is a Z line and this is the Z line, and the distance between these two Z lines is sarcomere. The space or the distance or the size of this A band is always fixed. A for always, A for all the time, A for any time. So anytime, all the time, always, the size of the A band is fixed. It is not moving. While the size of the I band, which is basically made of the actins, this is flexible. The size can increase and it can decrease as well. Normally, when the muscle is in a relaxed state, the actin molecules, they are separate from each other. They are not touching each other. They are not overlapping each other. But when the muscle is contracting, these actin molecules basically approach each other. These Z lines from one side and the Z line from the other side, they basically approach each other. And the actin from here and the actin from here, it may either touch each other or it may even overlap here. And you can see that the distance between the two Z lines or the sarcomere decrease, or the size of the sarcomere decrease during contraction. But during contraction, the size of the A band remains the same. As a whole, the muscle is basically made of small pockets of fasciculi. Each fasciculus is made of multiple fibers. Each muscle fiber is having multiple myofibrils, and while myofibrils is seen on an electron microscope, it is having dark bands and light bands. The dark band is known as A band, while the light band is known as I band. The I band or the light band has been divided into two parts by the help of Z line. The distance between two Z lines is known as the sarcomere. During the movement, the A band remains the same. The size of the A band remains the same, while the size of the I band keep on increasing and decreasing depending upon the movement of the muscle. If the muscle is in the relaxed state, the A band if the muscle is in a relaxed state, the I band, it will be its in its normal shape and size, and they will not be approaching each other. The Z lines will not be approaching each other, but when the muscle is contracting, the Z lines they basically approach each other. This actin basically they cross over. So the thick or the A band or the dark zone or the A band that is basically made of myosin. Here some actins are present, but this is basically made of myosin filaments, and this I band it is basically made of the actin. If we study in detail, in the relaxed state, when the actin from both sides has not approached each other, there is a small space between inside the A band, and this space is known as the H zone. This is the H zone. When the Z lines approach each other during the contraction of the muscles, this Z space, this Z zone, it gets diminished. You can see it has crossed over from both the sides. The Z lines have approached each other, and the H zone has basically disappeared. So this H zone or pseudo H zone, which is some books refer to it as pseudo H zone, it is only visible when the muscle is in its relaxed form. Now going into detail of the myosin and the actin. The myosin filament is basically made of myosin molecules, which is a protein. And this is basically a myosin filaments. This is basically made of myosin molecules. The myosin molecule in itself is made of 6 types of proteins. There are 4 light chains and 2 heavy chains. So you can differentiate the thick and the thin from the heavy and the light. The thick is having the myosin, the thin is having actin. Then the myosin is having heavy chains, 2 heavy chains and 4 light chains. The heavy chain is basically forming the tail of the myosin. This is a myosin filament. It has been taken from here. This myosin filament is basically represented here. It is basically made of multiple myosin molecules, and each myosin molecule is then made of 2 heavy chains and 6 light chains. So this is the tail of the myosin molecule, and this is the head of the myosin molecule. The head of the myosin molecules, it is basically making the crossover bridge. It is present here like crossover. This crossover bridges or the heads of the myosin molecules. It provides attachment of the myosin with the actin filament. The body of the actin molecule is basically formed of G actins, and these G actin combine to form the F actin. So the body of the actin molecule is basically made of F actin, and this F actin is basically made of G actin molecules. If we cut a muscle in its light zone or I band, they will appear like this. Only light chains will appear. But if we cut a muscle in a A band or dark zone, they will appear. The thick molecules will appear under the microscope, and it will be apparent that each thick or each myosin is surrounded by 6 thin or actin molecules. There are 3 other important molecules in the or important proteins in the actin. They are troponin, tropomyosin, and actin. So basically it's the actin filament. Basically, which is made of troponin, tropomyosin, and actin. The body of the actin filament is basically made of F actin, which is still made of the G actin proteins. Inside this myofibril, which is basically a part of muscle fiber, it basically behaves like a cell. So the space inside the two membranes of this myofibrils. This is known as sarcoplasm, and this is full of multiple ions like calcium, magnesium, sodium, potassium, etc. And these calcium, sodium, potassium, ATP or energy, this is going to help in the movement of muscles. This is going to help in the movement of muscle, in the movement of these actin over the myosin. So this is the basically the sarcoplasm. The space between the two Z line is sarcomere, and the protoplasm, just like the cytoplasm of a cell, this is it is having the sarcoplasm. Then apart from the sarcoplasm, this myofibril, it is also having a lot of mitochondria, and the more the demand for the movement, or the more the movement the muscle is doing, the more the number of mitochondria in that muscle. So let's summarize what we have discussed. The skeletal muscle as a whole, this is basically, when cut into two parts, you will see that this is basically made of multiple pockets of muscle fibers. Those pockets are basically made of known as fasciculi or fasciculus. Outside the muscle fiber, there is a thick connective tissue known as epimysium. The muscle fasciculus or the fasciculi are surrounded by a connective tissue known as the perimysium, and then inside the fasciculus or the fasciculi there are muscle fiber, and the muscle fiber are basically surrounded by the connective tissue known as endomysium. The endomysium connect with the perimysium, and the perimysium and the perimysium connect with the endomysium. They form the tendon, and tendon connects with the bone. So any movement in the muscle fiber or the myofibrils between the thick and thin filaments, they are basically transferred to the bone, and then the bone can be moved. So when we look at the fasciculus, you can see multiple mitochondria and these and the muscle fibers. When the muscle fiber is enlarged, it is basically made of the myofibrils. And when the myofibril is seen under the electron microscope, it is basically made of dark and light zone. The dark and light band, whatever. The dark band is known as the A band, which is basically due to its anisotropic structure characters, and the I band is basically the light zone is basically the I band, which is isotropic to light. The I band is further divided into two parts with the help of Z line, and one portion of the I band from either sides, it combines with the A bands to form the sarcomere. It is basically divided by the Z line. So the distance between two Z lines is the space is known as sarcomere. Then the dark zone is basically made of myosin filaments, and the I band or is the light region is basically made of actin filaments. The myosin filaments are basically made of myosin molecules, which are still made of 2 heavy chains and 6 light chains. It is having tails and the heads. The heads are basically making the crossover bridges to connect the myosin with the actin. The actin filament is basically made of actin, troponin and tropomyosin, and its body is made of, its basic structure is made of the F-actin proteins, which are made from the small G-actin proteins. We will discuss each and everything in the coming lecture, and the things will get cleared. In the relaxed state, there is a zone known as the H zone, and this is because the actin's filament from both the sides, it has not crossed over each other. So there is a light zone when the muscle is in the relaxed state. When the muscle contracts, and the contraction is because there is a head of myosin molecules, they pull the actins from both the sides, and the actin cross over each other, and the distance between the H zones, the Z zones decreases, the H zone disappear, the distance between two Z lines decrease while the H zone disappear, but the size of the A band remains the same. It is A for always, A for any time, A for all the time. All the times the size of the A band remains the same. So how basically it is moving, how basically uh, the inside the molecules, the ions like calcium, potassium, ATP, how they combine the heads of the myosin with the actin and how they help in the movement of muscles, that will be discussed in the coming lectures. Hope you have understood the starting lectures, and if there is any query, it will get cleared in the future lectures. Thanks a lot for watching.